Good day, grade 12s. Welcome to today's lesson. Today we're going to be talking about DNA, the code of life. This material is created by Thunder Eduk, Mr. M. Saidi. I'm using this material because it's the best material you can ever find out there. So let us start without wasting any time. Now, what is DNA? Now, DNA is often referred to as the code of life because it contains instructions on how to build various proteins. And these proteins will be important. We're going to see later on the importance of these proteins, but they're essential for existence. Now, before we get into detail, we have to revise the structure of the nucleus that we did in grade 10 in life sciences. Now, as you can see here, our nucleus has some organelles there that we cannot ignore. It has the endoplasmic reticulum surrounding the nucleus, and you can see this reddish dots all over the ER, and these dots are called ribosomes. And remember, in ribosome, we say this is where we're going to also talk about this important organelle. Now, in ribosome, it is where now the proteins are produced. Now, then we have the nucleus. Now, the nucleus, it is the important site, and it's the site where ribosomes are produced. Then we have the nucleoplasm. Remember, you also learned about the cytoplasm. Now, the nucleoplasm is also an empty space within the nucleus that other organelles are housed, such as the whole nucleus and these threads that we're gonna talk about in a few seconds. Now then we have this opening between here in the nucleus is gonna be called the nucleopore. Whatever that enters and exits the nucleus goes through this opening called the nucleopore. Then we have the surrounding of the nucleus, which is called now the nuclear envelope. Now, within this structure, then we have these threads. Now, these threads are called chromatin network. Now, if you see a nucleus like this, you can already tell that this nucleus is not active. But once it becomes active, this chromatin network will now undergo a process that we will see later on, DNA replication, where now they'll be preparing whereby the cell will be preparing itself for cell division. How can we tell that? We can tell that this cell is now ready for cell division by seeing this chromatin turning into visible chromosomes. Now, as you can see, a chromosome, this is what we also spoke about in mitosis in grade 10. A chromosome is made up of two chromatids held together by a centromere. Now, we're going to go into detail and talk about what really makes up a chromosome. Now, this is what we're going to be talking about during this lesson. Now, first, we have to talk about nucleic acids. And these nucleic acids consists of nucleotides. Remember, in grade 10, we spoke about organic compounds. And one of the organic compounds we spoke about was nucleic acids. Now, these nucleic acids that we're going to talk about now, we say they consist of nucleotides. Nucleotides, which are also referred to as building blocks or monomers, meaning that all the nucleotides are made up of all the nucleic acids are made up of many nucleotides bonded together by something or held together by something. That's when they are now referred to as building blocks or monomers. Now, each nucleotide is made up of a phosphate group, meaning that this group contains some phosphate or phosphorus ions. Then also a sugar portion. Now, this sugar can be deoxyribose or ribose. Now, this tells us in grade 10 we studied nucleic acids, and those nucleic acids are only two, which is DNA and RNA. Now, when you come here, when you see that the sugars are two, deoxyribose will be found in a DNA molecule and ribose will be found in an RNA molecule. Now, the third part that makes up a nucleotide is called nitrogenous base. And we have four nitrogenous base, which is adenine, thymine, guanine, and cytosine in DNA. But in RNA, we also have four, but the difference is that uracil will now replace thymine in RNA. These are the shape. The shape you'll always find that the phosphate group is represented by a circle and the sugar is represented by a pentagon. And a nitrogenous base will be represented by a shape of this kind. Of course, this shape will differ based on the base, but you will see later on. Now, this is the structure of a nucleotide. Look at it. C is made up of a phosphate group, a sugar portion, and a nitrogenous base. This is the structure of a nucleotide, and it doesn't change. The only thing that will be changing is the nitrogenous base that will be found here. It can either be adenine, thymine, guanine, cytosine, or uracil. This is the structure. It will always be like this. Then we look at types of nucleic acids. There are two types of nucleic acids. The first one is DNA, and DNA in full is called deoxyribonucleic acid. Then it is followed by RNA. RNA in full is called what? Ribonucleic acid. Then let's start talking about DNA. Now DNA, we have to know where it is found and its name. And here we are saying the types of DNA are based on where DNA is found. Let me repeat, the types of DNA are based on where it is found. So we name a DNA based on where it is found or based on its location. The first one, we have nuclear DNA, which is found in the nucleus. The second one, we have mitochondrial DNA, which is found in the mitochondria. The third one, we have chloroplastic DNA, which is found only in the chloroplast. And you can already tell, okay, all living things have cells. In animals, the cells will have all these organelles except chloroplast, meaning that in this scenario, an animal will have two DNAs. An animal will have a nuclear DNA and a mitochondrial DNA, whereas a plant will have all the three DNAs because chloroplast is only found in green plants. Now, the history of the discovery of the structure of the DNA molecule. This is for you to basically, understand the history, but it is not examinable. But you just have to know the significant scientists that contributed to the DNA molecule and the whole process. Now, James Watson, an American biochemist, and Francis Crick, an English physicist, began their collaborative work to try to solve the puzzle of the molecular structure of DNA. They used, now, there was like, what is DNA? How does it look like? Back then there were no enough material or resources to study things properly. Now, using data, specifically X-ray diffraction pictures provided by Maurice Wilkins and Rosalind Franklin, they made an accurate model of the molecular structure of DNA. That it is a double helix with a complementary base pair. This is what they discovered. Watson and Crick met with Wilkins. Wilkins showed Watson and Crick the X-ray picture of the DNA molecular structure. And then from there, Watson and Wilkins then concluded that DNA molecule, it is double helix and it has bases that comes in pairs. Now in 1962, Crick, Watson and Wilkins received the Nobel Prize for determining the molecular structure of DNA. By then, Rosalind Franklin had passed away. So that is why she's not mentioned in the people who won the Nobel Prize Award for this achievement. Now let us look at the structure of DNA. Now you'll be asked this question: describe the structure of DNA. This is how we approach such a question. DNA is made up of a nucleotide. Now, each nucleotide has a phosphate group, a deoxyribose sugar, and a nitrogenous base. Now, the nucleotides are joined together by a phosphate sugar bond to form a strand. Nucleotides after another nucleotide are connected by a sugar phosphate because the phosphate group attaches to the sugar. Then the sugar is attached to the nitrogenous base. Then the next nucleotide, same thing. There are 4 nitrogenous bases. In terms of DNA, we have adenine, thymine, guanine, and cytosine. And we use these letters to represent them. Now, these bases are complementary to each other. That is, adenine pairs with thymine, cytosine pairs with guanine. Now, nitrogenous bases linked by weak hydrogen bonds to form 2 strands. A DNA molecule is double-stranded, and the 2 strands are connected together by a weak hydrogen bond to form 2 strands. Now, when you say this one, you don't just say hydrogen bonds. You include weak hydrogen bond because when you just say hydrogen bond, you are now in chemistry. Here we say specifically weak hydrogen bond connects the 2 strands. Now, the 2 strands after connecting, they now twist to form a double helix. We'll see the picture later on. Now, a DNA structure we say is made up of a phosphate group. You can see there a phosphate group. Remember the shapes. A sugar portion. But this sugar now we call it a what? A deoxyribose sugar. Then it's made up of what? Bases. We have how many? We have 4 bases in DNA. We have adenine, thymine, guanine, and cytosine. And then adenine always pairs with thymine. Guanine always pairs with cytosine. They will go in that sequence in different ways to form a long chain, which now then becomes a DNA. So this is 2 strands. As you can see here, strand 1 and strand 2. And these bases are held together by a weak hydrogen bond. These lines are just showing us that there is a bond there that is holding. And you'll see, look at T. T, the shape of a T is inward. The shape of A is outward. And the shape of G is a circular inward. And this one is a circular. So a circular outward, a circular inward. From going down there, 1, 2 will be the same thing, AT, because the shape of A will be the same as the shape of another A. The shape of T will also be the same as the shape of another T. Therefore, if you see a circle outward, it will also be G. Here it will also be C. Then here it is now switched. This one is now T, this one is now A because of the shape. These shapes guide you so that when you have to identify, you look at those shapes and they will also guide you by giving you other bases. Now, the DNA structure is double-stranded. As you can see, it has 2 strands and it is held together by weak hydrogen bonds. And it contains a deoxyribose sugar and it has nitrogenous bases A, G, C, and T. And A is always complementary to T, meaning that A always pairs with T and G always pairs with C. Then it twists to become a double helix. Now, what are functions of DNA? Number 1, sections of DNA forming genes carry hereditary information, which is passed on from generation to generation. Number 2, DNA contains coded information for protein synthesis. All proteins that are produced in the body are produced as a result of the information that is found within the DNA molecule. Now, we have to understand the concept of base calculations, that as we say, adenine always pairs with thymine. Therefore, in a DNA molecule, the number of thymine will always be equal to the number of adenine. And the number of guanine will always be equal to the number of cytosine. And together, as for the they constitute 100% total. Meaning that if ever guanine is 25%, cytosine will also be 25%. Therefore, they all make 50%. Then adenine will also be 25. Thymine will also be 25. Therefore, together as 4, they make up 100%. But here, if A is 30%, what is percentage of C? A matches with thymine. Therefore, if A is 30%, then T is also 30%. Therefore, meaning that A and T constitute 60% of 100%. Therefore, you minus that 60% into 100, you minus it, you are remaining with 40. Therefore, that 40 is now divided between guanine and cytosine. Therefore, C will be 20. Guanine will also be 20. Will also be 20. We continue. These are similar other questions. You can pause here and try to answer this question and see if you get the picture. Now, the next concept is DNA replication. Now, the questions on DNA replication may come in this way. What is DNA replication? Now, DNA replication is a process by which DNA makes an exact copy of itself during cell division or before cell division. When in the cell does it take place? Now, DNA replication occurs during interphase, before cell division. So basically, interphase is not a part of cell division, but interphase, it happens before cell division, either before mitosis or meiosis, and it happens before any cell division, and we're going to see the importance of it. Meaning that during interphase, the cell prepares itself for cell division by doing what? DNA replication. Where does it take place? It takes place in the nucleus of the cell. And why does it take place? It's significant. Questions will come. What is the significance of DNA replication? Now, DNA replication occurs to double the chromosome number or the genetic material so that the resulting cells during cell division have enough or equal chromosome number. It also results in the formation of identical daughter cells during mitosis, because DNA replication, as we say, a process by which DNA makes a copy or exact copy of itself, meaning that the resulting chromosomes will be identical to each other. Now, let us explain the process of DNA. Now, DNA replication. Firstly, we say that we know that DNA is double-stranded and it is helical. This is the shape when you say it is a double helix. Now, during the DNA replication, the DNA molecule unwinds. It unwinds and starts to look like a ladder-like structure. Then the weak hydrogen bond between the nitrogenous bases breaks. Then the DNA molecule unzips or separates to form two separate strands. Now, so it forms two separate strands. Now, each strand will act as a template, whereby the information on each strand will be copied to make complementary bases of that strand. Then we're going to have two identical DNA molecules, and each DNA molecule will have an original DNA strand and a new DNA strand. Now, how does DNA replication take? As I've been explaining, DNA double helix unwinds or unwind. My learners love to make fun of me when I say unwind. Now, weak hydrogen bond breaks, and as it breaks, the two strands, they're similar like a zip of a gene. They unzip, they separate. to form two separate strands, each acting as a template using free floating DNA nucleotide from the nucleoplasm. Now, this nucleotide will be coming with a base that is complementary to the one that is on this DNA template. A complementary DNA strand is formed where A pairs with T and G with C. Two new DNA molecules are formed, which are genetically unidentical. They are not, sorry, I'm saying unidentical. Two new DNA molecules are formed, which are genetically identical, with one original strand and one new strand, as I've explained through that structure. Now, this whole process is controlled by enzymes, but you do not need to know the type of enzyme. But as a scientist, we must know that basically every reaction that occurs in our body is controlled by enzymes. And remember, enzymes are made up of what? Proteins. And then we get proteins from what? We get protein code from DNA. That is why now it is important to understand this whole concept. Now, we then go to the second concept, DNA profiling. Now, DNA profiling is a technique of identifying someone's unknown DNA profile using a known DNA profile. Then you can see here we have an example. We have a DNA sample that was found at the crime scene, and we have 3 DNA samples of 3 suspects who are thought to be one of the people who are involved in the crime. Now here we see these black bars. One will ask, what are these black bars that are shown on a DNA profile? Now, so these black bars, one will ask, what are the black bars representing DNA? Now, a DNA profile is a pattern of black bars that represent the DNA fragments of a person. So these are the fragments of a DNA of a person. Then if you get a DNA profile of someone and then you get another DNA profile, you're going to match the black bars. And if those black bars are matching, it's most likely that this DNA belongs to the same person. In this case, you have one from the crime scene. Now, I told my learners that when you're analyzing these samples of different people with one, you use a ruler. You use a ruler to place on top of all this, because they'll be in this order. You use a ruler to check which one is in the same line or is aligned or is matching. And as you use your ruler, you tick off those that do not match. You'll be left with one that matches. You'll be left with one that matches. You can see here, it matches. This one 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 matches. And this one, you can see that in this one is a crime scene. One suspect, 2 is a 100% match with the crime scene. A DNA profile found at the crime scene. So you can then now conclude that suspect 2 is the one who left his DNA at the crime scene. And then he'll have to explain how did his DNA get to the crime scene. So here in this one, to conclude that is suspect 2, you'll see by the number of black bars matching to the crime scene, the most numbers, because you can see this one there's 1, 2, but you'll be confused. Also 3, you'll think it's him, but when you compare suspect 2 and 1, 2 is the one who has the most black bars that match with the ones with the black bars of the DNA profile that is found at the crime scene. In this case, they will be identical because it's of the same. But then when you look at a paternity case, where now we are trying to confirm the father of the child's DNA profile will not be 100% matching with the father. Some black bars will be matching with that of the father, others with that of the mother. Now, what are the uses of DNA profiling? DNA profiling is used for personal identification, paternity and maternity. Paternity is testing for the father. Which one is the father in paternity? And maternity is maybe that relates to the mother. Maybe at the hospital, if there's a swap of babies, now they want to check who's the mother of this one. They now can conduct a DNA profile. The third one, diagnosis of inherited diseases. Usually, when people want to have babies, they want to see, they go for a family planning, they want to see what is the possibility of that child coming normal or in a certain way. Now, also, we use this one for criminal identification, as is in this case. Now, also, DNA profiling is also used in forensics for many things, just like even here it can also be done that way. Also, if maybe the police find a body that is bent beyond recognition or that is decomposed beyond recognition, they'll now be using DNA to prove or to find or to know possibly who's this person. Also, DNA profiling is used for identifying suitable organ donors. Now, there are also disadvantages of DNA profiling. Some complain that it is expensive. Not everyone has access to DNA profiling. It's people who are doubting that they are the father or yeah, it's most that one when you'll doubt and end up believing because you don't have that enough money to go and do DNA profiling unless you go to a television show and you seek help. Then also people complain it is analyzed by humans after all. So when they analyze it, they can make errors in analyzing. And also, not all hospitals have the necessary requirements to carry out DNA profiling because it's relatively new. It's not that old. In USA, it probably started in the 1970s. During that time, as you know, the police were now able to use DNA to pin or to get suspects in court. People were not really understanding DNA or liking DNA. Therefore, before proceedings, the prosecutors would normally call an expert in the DNA field who would come and conduct a lesson, teach the jury and the whole audience at large about DNA and why each and every person has a unique DNA. Therefore, so that people can now have that belief that DNA can now solve cases. Now, it is possible to plant DNA at a crime scene, resulting in a false evidence or an innocent person's DNA might be planted at the scene by someone, and then the police will think that that person is the guilty party. The guilty party. So people are like, they are skeptical. They say, you can't base convictions based on DNA alone. DNA can be planted. Once they find DNA at the crime scene, then you'd have to explain why your DNA is there. Or if someone is sadly killed and raped, then they can find the semen within that body, and you have to explain why is your semen in the deceased body and provide evidence if you had a